Hello, good morning. I hope you can all hear me fine and see my screen fine. If you can't, just type it into the chat box. Hey, good morning, everybody. Thank you for joining today's webinar. We're still waiting for some attendees to arrive, so we'll start in a few minutes. I hope that you can hear me fine, but if you can't, please just type this into the chat box. Thank you. Good morning, everybody. Thank you for joining today's webinar. My name is Malti Raman and I work as a product manager at Takara Bio Europe. And today I'll be presenting our webinar on Takara's next generation tools to clone, express and purify proteins to really help you speed up your protein expression and purification workflow. We will have time for questions at the end of the webinar, but if you have questions during the webinar, please just type them into the chat box and I'll answer them at the end. This webinar is also being recorded, so we will send you a link to the recording at the end of the webinar, as well as a copy of the presentation. Okay, so before I talk about our next generation technologies themselves, I first wanted to present you with a typical clone, express and purify workflow that most researchers use for their protein expression and purification experiments. With the first step being, of course, to clone the target gene of interest or protein of interest into your protein expression vector of choice. Now, most people are using traditional ligation-based methods for this step, but today I want to introduce you to our next generation infusion cloning system as a better way to clone. So after the cloning step, the next step is to then express the target protein, and this is usually expressed as a recombinant protein with a tag such as a his tag. And in fact, Takara offer a wide range of protein expression systems for this step, and today I'll just present you with an overview of these nice systems that we have. But you can find out more about this, more about these different systems on our website. So after expressing the target protein, usually with a tag such as a his tag, the next step is then to purify the target protein with this recombinant tag. And indeed, we also have our next generation Capture-M membrane-based technology, which is a much faster and easier way to purify proteins than using traditional resin-based methods, which is what most researchers currently use. Okay, so the first step is to obviously choose the expression system or vector that you want to clone your protein into, target protein that you want to study more. So as you can see, Takara, under both our Takara and Glontech brands, offer a wide range of different systems, including bacterial systems. So we have both E. coli and bacillus systems, as well as insect, mammalian, and even in vitro human cell-free systems. And as you can see, as you go from bacterial to human, the number of post-translational modifications that are possible also increase. Okay, so we really have a nice range of systems. So now I just want to present you in this slide just an overview table of these various systems to help you choose the right system for your experiment. So for example, if you're just looking for a simple low-cost system which gives you a good yield, then our first choice would be to try our E. coli PET express and purify system, okay, which is just a regular IPTG inducible protein expression systems based on the PET vectors. You can see that you get a good yield of protein. However, if you have a more difficult protein, which for example is forming aggregates or it just doesn't easily solubilize, then you could consider trying our P-cold vectors, which allows expression of your target protein under the cold shock promoter. So it's expressed at 15 degrees C. So this actually inhibits host protease activities and you end up with more soluble proteins. But as you can see, the yield isn't as high as the E. coli PET system. However, if you're really looking for a much higher yield, then you might be more interested in our bacillus system. So we offer both Brevibacillus and B. Cetillus systems, which actually allows you to obtain your target protein as a secreted active protein. So you can see the yields are much higher than the E. coli. So you can get up to 4 grams per liter with the Brevibacillus systems and up to 25 grams per liter with the B. Cetillus systems. And with both of these systems, you are able to have disulfide bond formation and also has very low endotoxin levels compared to E. coli. So you get a more functional protein being secreted out. So secretion is optional with the Brevibacillus systems, but with the B. Cetillus systems, in fact, secretion can also be optimized. So if you're really looking for a high yield system, I would recommend considering one of these. However, if the number of post-translational modifications is more important, then you could consider using our insect baculoviral system, which of course is closer to mammalian cells, and you still get a good yield. But however, if you really want to express in mammalian cells, we also have our HEK293 Ultra expression vectors, which allows you to directly express your target protein in mammalian HEK293 cells. But of course, you can see the yield is not as high as insect or the bacterial systems, but you'll get all the different post-translational modifications that you might need for your target protein function. And finally, we also offer our human cell-free systems, which is an in vitro transcription and translation system. So if you really have a toxic protein, you could consider using this system to express it in vitro. Okay, so once you've chosen your protein expression vector that you want to clone your target protein into, of course, you then need to do the actual cloning. So as I said before, most people are still using traditional ligation-based methods, which isn't always efficient and can sometimes take weeks to months to get your actual construct. However, we have a very fast and accurate cloning system called infusion, okay, which allows you to minimize the amount of time spent at this cloning step. And we really believe infusion is just a better way to clone. So my next few slides will talk about our infusion system. So infusion system, we really believe, is a cloning system without any limitations because it allows you to clone both single inserts and multiple inserts in a single step. It allows you to do site-directed mutagenesis in a single step and also easily clone N or C terminal tags in a single step. So you can do all these different types of cloning reactions with one system. It's really a cloning system with no limitations. You can design any construct and use infusion to deliver that construct. It's such a powerful system. So you might be asking yourself, what's so special about infusion? Well, the beauty of infusion lies in the fact that it's completely sequence independent. So it can be used to clone any insert into any vector at any locus. You're not limited either by the sequence of the vector or by the sequence of the insert. Okay? So this means that you can always directly clone into your final destination vector. You don't need to do any sub-cloning steps. And because you're not limited by the sequence of your insert, you're not limited by available restriction sites, which is a major limitation of ligation-based cloning. Infusion is also seamless, so you don't add any extra bases to your insert unless you actually want to. And again, this is a great advantage over other methods such as gateway cloning, where recombination sites are added to your target gene, sometimes resulting in additional amino acids being added to your protein of interest, which you don't want. Fusion is also directional, and you can also do some really nice applications like cloning multiple inserts and doing mutagenesis in a single step. Okay, so how does it actually work, this infusion system? So the first step is to linearize your vector. So this can be any vector that you have in your lab. So you're not limited to using any kit-specific vectors. You just need to decide where you want to clone your insert and linearize the vector at that site. And you can in fact linearize your vector either via restriction digestion if you have suitable sites, or if you want to be completely restriction site independent, you can also linearize your vector via inverse PCR. This is also possible as long as you use a high fidelity polymerase. Then you simply need to know the 15 bases found at either end of your linearized vector, shown in red and yellow here. And the same 15 bases are then added to either side of your gene of interest, which can be any gene. Okay, so you can either amplify your insert via PCR using gene-specific primers that also contain these 15 base pair overlaps to either end of your linear vector, or you can be even faster and just get it synthesized as a DNA, as a synthetic DNA fragment. As long as it includes the infusion overlap, you can use it with infusion cloning. After purification of the linearized vector and the insert, you then simply incubate it with the infusion enzyme for 15 minutes at 50 degrees C. And what the infusion enzyme does is it fuses the insert to the vector at these 15 base pair overlapping regions. When this is transformed into E. coli, gaps are repaired and you get positive clones containing your recombinant vector. It's a really simple and very efficient system. But of course, primer design is very critical here. And to this end, we have a new online primer design tool for infusion. So please always use this when you're designing primers for infusion cloning. So now let's take a closer look at the exact mechanism of the infusion enzyme. How does it fuse the insert to the vector? So if you imagine this is the insert and it already contains these 15 base pair overlaps to either end of the linearized vector, shown in red and blue here. So if you imagine that this vector is already linearized here, so this black region is missing. When you incubate the insert and the vector with the infusion enzyme's 3' exonuclease activity actually chews back the ends of the insert to the vector, allowing the insert to spontaneously anneal to the vector at these exposed 15 base pair overlapping regions. But at this point, the insert is annealed to the vector just via hydrogen bonding. It's only when this annealed molecule is transformed into E. coli that you get in vivo gap repair occurring. So gaps are repaired and you get permanent joining of the insert to the vector happening within the E. coli cell itself. Okay, this then results in positive colonies containing your recombinant vector. So what are the primary advantages of infusion over, for example, ligation-based method? The biggest advantage, of course, is cloning success. And this is because we've spent over 10 years of progressive research into perfecting this product. It allows you to clone directly into any vector. And this cloning is also seamless, so you don't add any extra bases to your insert, which is crucial when you're trying to generate protein expression constructs. And infusion really also has an excellent performance. So we routinely obtain over 95% cloning efficiency or positive recombinant clones for a wide range of insert sizes. So anything from 20 base pair oligos to 15 kb inserts. So this is really the advantage of infusion. So I think most researchers who use traditional ligation-based cloning screen at least 10 to 20 clones per construct, okay, before obtaining your positive clone. With infusion, due to the high cloning efficiency of 95%, you simply need to screen 2 colonies or 2 clones per construct, and both should be positive. So you can see you really save a lot of time and money on this screening step. And it's fast and accurate. It's just a 15-minute reaction. And it's simple and powerful. So you can use it for large inserts, for single, or even for multiple inserts. You can see here in this graph that we have used infusion to clone 8 kb, 12 kb, and 15 kb inserts. So as you can see, although the total number of colonies does drop as you clone longer inserts, and this is because you're putting more pressure on the cells with longer inserts, the percentage of positive clones remains high at 90 to 100%. So when we screen 10 clones by colony PCR sequencing, you can see either 9 out of 10 or 10 out of 10 were correct. So it just illustrates the power of infusion. And it's really one system that can be used for multiple applications. So you can really use infusion for all your cloning experiments. Infusion cloning is also accurate, directional, and seamless. So we know that sequencing data, of course, is a gold standard measure of cloning accuracy. So here we have some sequence data from positive clones generated with infusion. So you can see firstly, sequence is error-free. So this illustrates that the polymerase that we provide in our infusion kit called CloneAmp is very high fidelity. You can see that the cloning is also directional, and also that there are no bases in between the vector or insert, so it's really seamless. So in this way, you don't need to, screen many colonies at all, and you also don't end up with any unwanted amino acids. Infusion is also very versatile and can be used for many different applications, including, so apart from single fragment cloning, you can use it to clone into large vectors. The largest vector that we've cloned into has been a 42 kb cosmid vector. You can also use it for multiple fragment cloning, so really just to assemble multiple inserts in a single reaction. You can use it for site-directed mutagenesis. You don't need a separate kit for this. You can also use it for CRISPR-Cas9 cloning and for adding small N or C terminal tags. Really, you can design any construct and you use infusion to generate this construct. So I won't have time in today's webinar to talk about all these different applications, but I will just focus on multiple fragment cloning and site-directed mutagenesis. But you can find out more by visiting our website for these other different applications. Okay, so let's first look at multiple fragment cloning. So this is a really nice application of infusion because it allows you to assemble multiple inserts as if it was a single insert in a single reaction with infusion, and you're not limited by the sequences of the different inserts. So if you wanted to assemble multiple inserts via ligation-based cloning, you would have to do several rounds of subcloning, whereas you don't need to do this at all with infusion. So how does this work? So the first step is, of course, to linearize your vector at your desired location. So this can be, again, via restriction digestion or via inverse PCR. The next step is to then amplify, or you can synthesize your different inserts that you want to clone. So in this example, we've shown 3 different inserts shown in red, blue, and green here. So if you amplify the inserts via PCR, you just need to make sure that each insert either has a 20 base pair overlap to one end of the linearized vector or to the adjacent insert to which it will be joined. So you can see that primer design is critical here. And so I would recommend you to use our new online primer design tool to design these 20 base pair overlaps. And the reason we recommend using 20 base pair overlaps rather than 15 base pair overlaps for multiple insert cloning is because this results in a higher number of total colonies, because of course as you clone more and more inserts, there's more and more pressure on the cells, so the total number of colonies does drop as you clone more and more inserts. But by using 20 base pair overlaps, you are able to still get enough colonies versus 15 base pair overlap. However, we still recommend just 15 base pair overlaps for single insert cloning because this is optimal. So one way is to amplify the inserts via PCR, but you can also be faster and just get them synthesized as synthetic DNA fragments. As long as you include the required 20 base pair overlaps for infusion, this is fine. And then you simply incubate the various inserts and the linearized vector with the infusion enzyme. So the infusion enzyme, as before, just creates single-stranded regions at the ends of each insert and the ends of the vector, allowing them all to spontaneously anneal at the exposed 20 base pair overlapping regions. So in this way, they're effectively cloned as a single fragment using infusion. And don't just take our word for it. We have a nice paper for this. And another related application, so for those of you who do synthesize your genes for cloning or synthesize your inserts for cloning, if you have problems with synthesizing longer inserts, such as inserts over 2 to 3 kb due to secondary structure issues, you can get around this issue via multiple fragment cloning because you can simply split your longer gene into smaller fragments, okay, and then simply use infusion to assemble the smaller fragments. So we have a nice tech note to explain this, so how infusion can be used to solve this long DNA synthesis challenge. Now I just wanted to show you some in-house data that we have for a multiple fragment cloning reaction using infusion. So here we cloned 3 different inserts which were PCR amplified. So these inserts were either 1.1 kb, 0.7 or 0.7 kb, and they were all cloned using infusion into a linearized 2.7 PUC19 vector to get a final construct of 5.2 kb. So after the 15-minute at 50 degrees C infusion reaction, you can see that we obtain 89 colonies in total. And out of these 89 colonies, when we screened 26 for the presence of the insert both via colony PCR and by sequencing, you can see that 26 out of 26 were correct, leading to 100% cloning efficiency or accuracy, which is a great result for a multiple insert cloning reaction. So for those of you who are assembling multi-protein complexes or doing transgenic constructs, this is a really nice application of infusion that can help you generate these constructs more quickly. And you can see we also just obtained one colony on the negative control plate, which shows that there's very little background coming through in this reaction. Another really nice application of infusion cloning is that you can also use it for mutagenesis. So you can do both deletion, substitution, and insertion mutagenesis. I'll just talk about substitution mutagenesis today because I don't have time to talk about all the different mutagenesis types, but you can find out more by going online or reading our Nature Methods app note shown in the link here. Okay, so how can you do this substitution mutagenesis with infusion? So if you imagine that you have a vector with your insert already cloned into it, so this is shown in yellow here, right? And you just want to alter a few bases within your insert. Now all you need to do is simply linearize this vector via inverse PCR, but you use primers where you incorporate the bases to be mutated within the 5' region of each inverse PCR primer. So these are shown in as X's here. But the 5' end of each primer also has a 15 base pair overlap with each other. So in this way, when you obtain your linearized vector, okay, the ends of the vector not only have an overlap with each other, but also have the mutated bases. And then you simply use infusion to recircularize this vector with the mutated bases using infusion. Okay, because of course you have the overlaps required for infusion. So it's a really nice application of infusion. So you don't need a separate kit for mutagenesis. So the infusion system that we currently sell is called Infusion HD Cloning Plus, which we believe is the cloning solution to all your experiments. So what comes inside the box is, of course, the infusion enzyme, but we also provide a high-fidelity PCR polymerase called CloneAmp Hi-Fi. It has one of the highest fidelities on the market and is ideal for cloning with infusion. Our Plus kit also comes with a choice of insert purification kit, so either spin columns or something called cloning enhancer if you have pure PCR products. And lastly, it also comes with our stellar competence cells, which are very high competency cells, which are ideal for use with infusion cloning. So really, we provide all the optimized subcomponents of infusion in our Plus system. And we recommend using this for the best results with infusion. But equally outside of the box, we provide expert tech support. We're continually improving our product and services to ensure our customers' success. And in fact, our Plus system is really a value pricing, because if you were to buy these components separately, it would be much more expensive than what we offer as our Plus kit. And don't just take our word for it. We have many papers out there that have cited in the use of infusion for their cloning experiments. In fact, we have over 1850 publications. Okay, so that was our infusion system. So once you've generated your protein expression construct containing your target gene, the next step is then to, of course, express your target protein. So I already presented you with a quick overview of the different systems that we offer at Takara, but I just wanted to go into detail about the B. subtilis and PHEK293 vectors that we offer, because they're a bit more specialized. Okay, so with the B. subtilis system, you can really optimize secretion of your protein. So if you're really looking for a high yield of your target protein, this would be the best system, because with our B. subtilis system, we provide you with a signal peptide library. So we provide 173 different signal peptides. So the idea is that with our system, you generate a library of your target protein, but with different signal peptides to see which signal peptide leads to the highest yield of your secreted protein. And in fact, we also include infusion within our system because it's used for this library scale SP cloning. And it's just a really nice system because it's also endotoxin free, it's a safe organism, so you can really achieve medium to large scale production of your proteins or enzymes of interest. So it's something which is quite unique to Takara. Another quite unique protein expression system is our ultra high expression PHEK293 vectors. So if you're looking for vectors that allow high level protein expression in mammalian cells, this could be of interest to you. So these are vectors that basically have these HIV1 TAT and TAR elements, okay? And you get high level protein expression via this HIV1 based TAR TAT transcriptional activation. So the idea is that you clone your gene of interest into this vector, of course, using infusion for the best results, and then you just transfect this vector into your into 293F suspension cells. So once you've transfected, you get this mRNA being produced with this TAR element being like a hairpin loop at the 5' end of the RNA, which then activates TAT. Okay, TAT then binds to TAR, and then you get transcriptional activation via TAR, which allows high-level expression of your protein of interest. And in fact, we have a nice online tech note where we used these vectors to express monoclonal antibodies, so you can find out more here. So we have two different types here. So vector 1 has both the TAR and TAT on the same vector, whereas in expression vector 2 system, these are provided on separate vectors. So one vector has TAR, one vector has TAT. That way you can more closely tune the level of TAT expression. Okay, so once you've expressed your target protein, and most researchers usually express the target protein as a recombinant protein, the tag such as a his tag, the next step is then to purify the target protein, making use of this tag. And to this end, we have our next generation capture and membrane-based technologies to really speed up your target protein purification. So we have capture kits for both his tag protein purification and for native antibody purification. And this is much faster than currently used resin-based methods, and also much easier. And I'll talk about this in my next few slides. So as I said, we provide capture technologies for fast and easy protein purification of both his tag proteins and untag antibodies. Okay. So our capture membrane is basically, it's a high capacity membrane which either contains nickel, immobilized nickel for his tag protein purification, or it contains immobilized protein A or G for antibody purification. So we have all these three types of kits. And in all cases, we have assembled these membranes into spin column or 96-well plate formats, okay? So that it's now, you can now do like a his tag protein mini prep. It's as easy as doing a plasmid DNA mini prep. So we have for all these three versions, a mini prep format, a maxi prep format, and a 96-well plate format if you really have many samples to process in parallel. And the 96-well plate can also be attached to a vacuum manifold. Okay. But the advantage of our capture technology is it's just very fast. It's really as easy as equilibrating the column, binding your sample, washing and eluting. And with the mini prep format, this entire process can be completed within 5 minutes. And this goes up to 15 minutes with the maxi prep and 96-well plate formats. So you can see it's just much faster than resin-based methods. And the other advantage is it's also compatible with many different additives and lysis buffers. And I'll talk about this more in my next few slides. So you might be asking yourself, so what is this capture membrane? What's so special about it? So let's take a closer look at the membrane technology compared to resin-based columns. So if we could first consider a resin-based column. A resin-based column has a high capacity due to a large bed volume. Okay, but the disadvantage of a large bed volume is that you get a slow diffusion of macromolecules through the pores, and this leads to long separation times. And indeed, at Takara, we do offer some resin-based products for his tag protein purification, his 60 nickel and talon resins. But now our R&D have invented this new Capture M high capacity modified membrane. So here it's a membrane where the pores have been chemically modified to have a higher surface area or a higher capacity than traditional membrane pores. So as with traditional membranes, you get a much faster purification than resin because, of course, of the small bed volume and rapid flow, but you get a better yield than traditional membrane pores because of these chemically modified pores, which have a higher capacity. And as I said before, we have our Capture M kits for histag purification and also antibody purification. So this is just a close-up of the Capture M membrane, how it looks. Okay, so first I'll talk about our Capture M histag purification kits. So the protocol is very, very simple, and it's essentially the same for our mini prep, maxi prep, and 96 watt plate format. It's just the spin time is longer for the maxi and 96 watt plate formats than the mini prep. Okay, so let's go through the protocol step by step. So the first step, of course, is to express your target histag proteins in your target cells of choice, and then to prepare clarified lysate from these cells. So these can be bacterial cells or mammalian cells, or you can also directly load cell culture supernatant. So for example, if you have a secreted protein, you can also directly load this onto our Capture M membrane. So once you've prepared your lysate and you've equilibrated a Capture M column, you then simply load this clarified lysate onto the equilibrated column. And the beauty of Capture M is, because it's such a thin, flat membrane, you get immediate binding of your histag protein to the membrane. So there's no incubation step here, which is where you save time compared to resin-based columns. But it's important to clarify the lysate, because if you have any debris left, it can clog up the membrane at this point. And another advantage is if you have dilute samples, you can also do several loads of your sample. As long as you don't exceed the maximum binding capacity of the membrane, this is fine. Okay, so after binding, you then wash. And because of the small bed volume of the Capture M membrane, this ends up being a high volume wash, which removes most contaminants. So it's much purer than resin-based methods. And then you also elute in a small volume because of the small bed volume. So in this way, you can not only concentrate dilute samples, but you also end up with a more concentrated eluate than traditional membrane-based methods. So when we analyzed our different fractions, so here we expressed histag GFP UV, and then used our Capture M histag mini prep kit to purify this. You can see that we got nice clean bands here in the first elution fraction, and nothing was lost during the wash, so it works quite nicely. And in terms of the yields that you can get, with the mini prep format, the maximum yield that can be obtained is 100 micrograms of histag protein per column. And this is the same for each well of the 96 well plate. And it goes up to 2.5 mg per column for the maxi prep format. So depending on the yield that you need, you could consider using any of these three formats. But the advantage, of course, is that you get a very fast purification. So if you're screening lots of protein expression constructs in parallel, this could really save you time. Now, I also wanted to talk about a new product that we've launched in the last few months called our Capture His Tag Purification large volume kit. So it's a large volume version of our Capture His Tag kit. You can see in this large volume version what we provide is this filter unit. Okay, so the capture membrane's here, but the filter unit can be attached to a vacuum pump, and you simply load your sample into the filter unit and then apply the vacuum to drain the sample through the membrane. Okay, so here we actually used this filter unit to purify a secreted His tag metrodioluciferase that we expressed in 293 cells. Okay, so we simply loaded the cell culture supernatant containing this His tag metrodioluciferase into our filter unit. So we did 2 lots of 600 ml loads. Okay, so this was after it was equilibrated. Then we did a wash and an elution. So you can see the elution volume is fairly small. And all this was done within 30 minutes. So you can see it's much faster than doing this kind of large-scale purification using resin-based methods. And here, although we show cell culture supernatant in this example, you can of course also load up to 500 ml of clarified bacterial or mammalian cell lysate as input. This also works fine. But again, it's very important to clarify to remove any large debris that could clog up the membrane. But in this way, you can purify up to 25 mg of His tag protein per unit. So this is pretty good, and it's very fast compared to resin-based methods. Now the other really great advantage of our Capture membrane is that it allows the use of additives, okay? And this is because the membrane, the bed volume is so small that the protein doesn't sit on the membrane for very long, so it is quite tolerant to many different additives such as EDTA, beta-mercaptoethanol, TCEP, and DTT. So EDTA is a common protease inhibitor that's used during protein purification, but usually it degrades resin. So if you use EDTA, you first need to do a buffer exchange before doing your resin-based purification. Whereas with our Capture columns, you can use even up to 10 millimolar EDTA with no effect on the protein purification at all. And similarly, you can use up to 30 millimolar beta-mercaptoethanol. You can use up to 10 millimolar TCEP and also 10 millimolar DTT. So it's quite tolerant of different additives, and these are other additives that we've also tested. Okay, so the structure of the Capture membrane also makes it ideal to purify difficult proteins such as, for example, membrane proteins such as G protein coupled receptors. The G protein coupled receptors are difficult to purify because of low expression levels, high hydrophobicity, so they're often not soluble, and also low stability in detergents. So often to make them soluble you need to use detergents, but then they have a low stability. So all these 3 points make these proteins difficult to purify. However, our Capture membrane can allow us to overcome all these 3 different issues. So for the low expression levels, often researchers get around this by expressing the protein in a high volume to get sufficient protein. So if you're using resin-based methods for this step, you often need to use a high volume of resin, which is expensive. But also you end up with a dilute protein, okay, so which in a larger volume, which may need to be concentrated afterwards. Whereas with our Capture Maxi Prep or large volume kits, you can pass a high volume of lysate. Remember, you can do several loads of dilute samples, but you still end up purifying a concentrated protein because of the small bed volume of the Capture membrane compared to resin-based methods. Okay. And equally, in terms of high hydrophobicity, of course, most researchers get around this issue by using detergents or other additives to make the protein soluble. But often these additives are not compatible with resin. They often degrade the resin. So often researchers first need to do a buffer exchange to remove the additives before doing the actual purification, Whereas with our Capture M products, because it's fast, because the protein doesn't sit on the membrane for very long, you can directly use the additives and you don't need to remove them before purification. And you still get a concentrated protein at the end. Finally, in terms of low stability in detergents, again, because the Capture M technology is so fast, the protein time in detergent solution is minimized to minutes, so it's not so affected as it would be with resin-based methods, and you get an improved protein reconstitution and functionality after purification compared to resin-based methods. So again, this is another way that the structure of the Capture M membrane allows the purification of such difficult proteins. So that was a theory, but and I now want to just show you some data where we actually used our Capture M His Mini Prep to purify a His-tagged GPCR, namely His-tagged ADRB2 or adrenoreceptor beta 2. So we expressed this protein in 293T cells, okay, prepared lysate from those cells, and then used our His Mini Prep to purify the His-tagged protein. Okay, so after STS page and blotting onto a membrane, we stained the membrane with Ponsu. You can see that the protein is present in all the different fractions. Then when we did a specific Western blot with an antibody against ADRB2, you can see that the protein is enriched in the iluate. So in this way, we were able to purify this His-tag GPCR within 5 minutes using our Capture M His Mini Prep. And finally, I just want to also talk about our Capture M Protein A and Protein G kits for antibody purification. Essentially, the concept is very similar, that you have this high-capacity Capture M membrane, but instead of containing immobilized nickel, our antibody purification kits either contain immobilized Protein A or immobilized Protein G. Okay, and immobilized Protein A or G actually binds to the Fc portion, the fixed constant region of antibodies, and this way you can purify antibodies easily using our Capture M Protein A or G columns. Okay. So I just want to show you some data on the Protein A kits, but you can find out more also about our Protein G kits, which are very similar on our website. Okay, because the principle is essentially the same. So as with the His kits, the first step is, of course, to prepare your antibody-containing sample, which can be serum, it can be cell culture supernatant, or clarified lysate. It's really up to you. It's just very important to dilute this in our recommended binding buffer, because this brings your sample to the same pH as a membrane, which allows a better binding of the antibody to the membrane. So again, you load this onto an equilibrated Capture M column, and as before, you get immediate binding of the antibody to the membrane because it's so thin and flat. So you save time at this step because you don't need to optimize binding here. You then do high volume wash, so you remove most contaminants, and then elute in a small volume. So in this way, you end up with a pure and concentrated antibodies. And again, with the Mini Prep protocol, this takes just 5 minutes, and it goes up to 15 minutes if you use a Maxi Prep or 96-well plate format. As you can see, when we run the different fractions on an STS page, you get really nice clean bands for the antibody. So this is the heavy chain band, this is the light chain band, with very little albumin coming through. And this slide just shows our Capture mini spin columns ability to bind antibodies in different animal sera. So you can see we tested sera from sheep, goat, rat, mouse, human, rabbit, and horse, and we were able to purify antibodies from all these different animal sera. You can see the bands are very clean, but the yield is dependent on the species and the isotype of the antibody. So these are the maximum yields that can be obtained with our Mini Prep 96-well plate Maxi Prep format. So you can see you can get up to 300 micrograms of antibody per column for the Mini Prep format. And it's the same per well of the 96-well plate, and it goes up to 2.5 mg for each column of the Maxi Prep format. But of course, this really depends on the species and the antibody isotype. Another nice application of our Protein A and Protein G kits is that it can be used for rapid immuno and co-immunoprecipitation. So not only can you purify the antibody, but you can actually also purify the target protein of the antibody. Okay, so how does this work? So you can immunoprecipitate the target protein of your antibody. So you simply just need to first prepare lysate from target cells expressing your protein, okay, and then incubate it with a polyclonal antibody against your target protein. Okay, you incubate this in a separate tube, Eppendorf tube, for 10 minutes with end-to-end rotation on a wheel, and this can be done at room temperature. This is then applied onto an equilibrated capturum column, so it could be protein A or protein G column. So this is fine. So again, you get immediate binding of the antibody to the membrane, but you will also get co-purification of the target protein bound to the antibody. Okay, so we did this with the PPTA-B subunit antibody. So here we used a polyclonal antibody with lysate expressing this protein, and then purified via our capturum protein A columns. Okay, and then when we did an SDS page and Western blot with a specific antibody against PP2A, the different fractions, you can see that it's enriched in the iluate here. So this is a really fast IP protocol. You can do this within 20 minutes. This is much faster than traditional immunoprecipitation experiments, which most researchers do with resin-coated magnetic beads. So often you first need to bind the magnetic bead to protein A or G, and then to bind it to the antibody and then to your sample. So it's a much longer process than with our capturum columns. And similarly, you can also do co-IP experiments. So you will not only pull down the target protein of the antibody, but also any bound interactors of the target protein. So we did this experiment to purify P53 or co-immunoprecipitate P53 and SV40T large antigen, which are known strong interactors. So here we first prepared lysate from 293T cells, which we know endogenously express both SV40T antigen and P53. Okay, we then did the initial IP with a polyclonal antibody against SV40T antigen. Okay. And then did the rest of the protocol as per here, and then we did a Western blot with a monoclonal antibody against SV40, and then we stripped and reprobed the same membrane with a P53 monoclonal antibody. So you can see that in the different fractions here. So this is the original sample flow through and elution fraction. You can see that we were able to pull down both in the elution fractions, and it's enriched here. Whereas they weren't, we didn't pull them down in the negative controls, which where no antibody was added for the initial IP. So it's a really nice, fast IP and co-IP protocol. And now we actually offer a capturum IP and co-IP kit, which comes with our protein A columns and specific buffers as well. Okay, so that brings me to the end of the webinar. You can find out more about infusion or our capturum technologies or our other protein expression systems by visiting our cloning resource portal or our protein learning center on our website. Or you can also email us at tech, so techeu@takarabio.com. So if you have questions, I can take them now. Just type them into the chat box if you have any. Okay, there don't seem to be any questions, so thank you again for joining us this morning. And if you think of any questions afterwards, okay, sorry, we have one question. Can you please repeat the principle of infusion cloning? Okay, yeah, sure. So let me just go back to my slide. Okay, so the infusion cloning principle just is based on the fact that you just need to know the 15 bases found at either end of your linearized vector. So you can use any vector, just linearize at your desired locus that you want to clone your insert into. So you can either do this via restriction digestion if you have suitable sites, or via inverse PCR if you just want to be completely restriction site independent. Both are possible. But once you have the linearized vector, you just need to know the 15 bases found at either end. So this is just shown in red and yellow here. And the same 15 bases then need to be added to either end of your insert. So this can again be any insert, okay? So once the insert and vector have the same 15 base pair overlapping regions, and they're incubated with the infusion enzyme, what the infusion enzyme does is through its 3' exonuclease activity, chews back the ends of the vector and the ends of the insert, allowing the insert to spontaneously anneal to the vector at these exposed 15 base pair overlapping regions. Okay, so that's how the enzyme works. But at this point, the annealed molecule is just held together via hydrogen bonds. It's only when you transform this into E. coli that you get in vivo gap repair occurring. So gaps are repaired and you get covalent linking of the insert to the vector happening in the bacterial cell itself. And then you get positive clones containing your vector. Okay, let's see if there are any other questions. Yes, I think I answered the second question, which are how are gaps repaired after insertion. So the gaps are repaired actually in vivo in the bacterial cell itself using the cell's own machinery. And we recommend, of course, our stellar cells for the best results, but you can use other cells such as DH5 alpha if you wish. They just need to have a high competency. It needs to be over 1 × 10⁸ CFU per microgram of DNA. Okay, yeah, so I think I explained the principle of the enzyme. Okay. Okay. Great. Okay, so I think there are no more questions. So thanks again for joining us. So this webinar has been recorded, and we will send you a link to the recording as well as a copy of the presentation by email. So I wish you a nice day. Thank you.